This is a production of Cornell University.
Okay, so again, thank you so much for the invite for myself in Virginia. And I'm going to start our discussion here and turn it over to Virginia in a few minutes. So we're going to talk about genetics, transgenics, and speed breeding to restore the American chestnut. And this first slide here, I think kind of encompasses quite a bit of what we do. Here in the left photo, you see a chestnut seedling. That's one of our transgenic crosses that's got an extra gene for blight tolerance. We'll explain how we do that in a few minutes. And so in our greenhouse, then we're producing pollen, male pollen, for use to take it to the field. Here on the right side of your screen, you see one of our 4 big wild type chestnut trees in Maine. This is a tree about 40 years old, maybe 50 feet tall, that we've used over the past couple of years for crossing with our transgenic pollen. And you can see a characteristic form of this tree that indicates it's suffering from the accidentally imported fungal blight. You see this big cluster of branches and leaves here on the bottom, indicating there's fungal blight up above in the stem and in the branches, and it's trying to compensate and grow below the fungal blight. So that's sort of telltale sign of what the problem is out in the field. Okay, so we're going to talk today about the progress we've made at the University of New England, UNE, but I want to acknowledge our colleagues, our scientific collaborators at down the road from you in Syracuse at the Environmental Science and Forestry College. The 3 individuals here in this picture on the right, Bill Powell, who's the person who's actually inserted the wheat gene into the chestnut that gives it blight tolerance, Allison Oakes. And it's interesting, so she did her PhD on chestnuts, but she's now working on the elm tree and restoring the elm tree based on the work she did with chestnuts in working on elm yellows. And then Andy Newhouse, also super important to the project and one of our major collaborators. So we're going to talk about our restoration science today, but we find sometimes humanists can tell the stories so much more powerfully than we can. So we're going to turn over just to a real quick 2-minute video produced by a documentarian, a videographer here at UNE, who has really latched onto our story and is excited about it and put together this little video that I'm going to share with you right now that kind of gives you an overview of what we're up to. Okay, so turn over to this. We're here doing a major ecological restoration project where we're making some great strides today in the direction of bringing back the American chestnut tree to the eastern United States.
So what we're doing is go up in the lift, we find the female flowers called the burrs, and we prepare them by clipping off the male catkins and put a protective bag around them so there is no open cross-pollination from other chestnuts in the area.
We pollinated the first Maine wild chestnut trees ever that were pollinated with pollen that we created in our greenhouse, in our lab, that has blight tolerance. So this pollen has an extra gene that allows it to withstand this invasive fungal blight that has wiped out virtually the entire population of the American chestnut tree throughout its eastern range from Maine to Alabama and across to Indiana. We collected the fertile nuts. We tested them to see if they've inherited the gene for blight tolerance. Close to about 47.5% of them did, which is what you'd expect because of the genomics. And now we are beginning to grow those out in our greenhouse. And we've also exchanged our chestnut seeds that have the blight tolerance with our partners throughout the eastern United States.
With partners like the University of New England, we're able to broaden everybody's horizon by working both interchapter and having the expertise of the university.
So here in Cape Elizabeth, we have the first field experiment ever in New England, where we are planting the American chestnut trees that have an extra gene for blight tolerance. So we know from lab experience that the extra gene protects the tree from the fungal blight. Now we need to see how it performs in more natural conditions. And this is the first time this has been attempted in New England. And so our Cape Elizabeth site is very important for the future of the American chestnut tree. Okay, trying to get out of this screen. Okay, let's see if I go over here. Okay. Okay, so we're back to the PowerPoint on your end. Want to make sure. Okay. Okay, so hopefully that gives you a bit of a flavor for what our project's about. And so here you see the native range of the American chestnut up till about 1900, when the fungal blight began to cut away at the population, of which reached close to 4 billion trees at its peak. And this fungus is Craphinetria parasitica, which has Asian origin and was accidentally imported with the Asian species in the late 1800s. So Craphinetria parasitica, what does it do to the American chestnut tree? Well, it finds fissures in the bark, weaknesses in the chestnut bark, and penetrates to reach the living layers. On saplings, you can see it can be quite aggressive. This orange indicates the fungal blight surrounding the stem and killing the tree above the stem. It does not, the fungal blight does not kill the chestnut tree below the ground. The microbes protect it. And so what you see happen then is that you see these root shoots coming up and from the root side of the tree. And so you have sort of a functionally extinct species that's still present often on the forest floor, but does not reproduce, does not play its ecological role. And then on the right, you see what the fungal blight looks like microscopically. It spreads both through the air and through squirrels and birds. And it's ubiquitous, so it's not going to go away. So the solution is not to eliminate the imported fungal blight, but to try to deal with it and try to reduce its toxicity. So much work has been done, and I'm going to just say a few words about a more traditional approach, more breeding approach to trying to deal with the fungal blight. And that's involving co-breeding or crossing the Chinese chestnut, which is Castanea mollissima, with the American chestnut, which is Castanea dentata. And you see from historical genomic analysis that the two species have been separated for about 20 million years. In the mollissima, the Chinese co-evolve with the fungal blight. So it tolerates the blight. The blight reproduces on the mollissima, but it doesn't kill it. So it functions as a saprotroph. In the context of the United States, where the chestnut here, this native species, has no experience prior to this accidental importation, the fungal blight acts as a necrotroph. So it kills the American chestnut tree, but it doesn't need to. It can function quite fine, as it does in Asia for millions of years, as a saprotroph. Okay, so then the idea has been in the breeding program to take the important genes for blight tolerance from the Chinese tree. You can see it depicted here on the right. It's a low kind of spreading tree, more of an orchard-shaped tree. The American chestnut is a tall canopy tree that reaches over 100 feet tall. So you see they're very different phenotypically. So that the approach has been to try to bring over what's good about the Chinese genome, but retain the phenotypic form of the American chestnut, and that produces what we call an F1, a 50-50 hybrid. So they do interbreed. There is some sterility, but despite the millions of years of separation, they can reproduce. But you produce something that's not competitive in the forest because of its size, and of course it has way more genes at this stage than you'd want to restore a native species to the eastern US forest. So what has been going on in breeding then is to select generation after generation of these crosses that phenotypically look like they have a substantial amount of blight tolerance. And prior to the current genomic revolution that we're fortunately part of today, and we're learning so much from, the thinking based on these generations of breeding was that it was only 2 or 3 Chinese chestnut genes that mattered, that needed to be moved over. So it looked like a fairly doable breeding project. And then eventually the idea after 6 generations, and this takes decades to do in the field, the idea would be that you'd have a tree that had high levels of blight resistance or tolerance, which was hoped, but that has not turned out to be the case. Why? Well, again, because of genomics, we can look now at the genomic profile of the chestnut tree. The chestnut tree has 12 chromosomes, and look at, well, where are the parts of these chromosomes that really matter, that show significant loci for blight tolerance. And these are, I won't go into a lot of detail on this, but there's 2 different major lines of Chinese trees that have been used in that breeding program, the Clapper line and the Graves line. And you can see wherever you see the line that bumps up, it's correlated with phenotypic results that indicate relative blight tolerance. And so you see that the basic point is that the blight tolerance is scattered across at least 9 of the chromosomes. You see that the 2 lines are correlated, but not perfectly. So there are different things coming over from each of those lines. And so the major challenge of this breeding approach is, how would you bring over the important loci, and how many do you need to bring over? This is work that other scientists are working on, but you can see it's a tremendous challenge. What we're doing is a different approach in that there is a common defense amongst plants to fungal attack, not only the Crophinetria parasitica that attacks the chestnut, but other types of fungus. And that's to utilize an enzyme, naturally occurring enzyme called oxalate oxidase, or what we abbreviate as OXO. And this is widespread amongst plants. And here's an example of a variety of different wild plants that have the OXO enzyme, and a whole bunch of domesticated plants that also have the enzyme. So it's likely that you, in the last few days, consumed this enzyme, you know, if you've eaten barley or banana or strawberries or rice or wheat. Wheat is the one that our work features, mainly because wheat is the most commonly studied genome, given the history of human domestication of that particular plant. So we know more about wheat than we know about cacao. And so that's the one that was selected. And the gene has then been inserted into the American chestnut, and that's creates this transgenic chestnut. And then just sort of to show you the widespread OXO gene in so many cultivated foods here on the right, we keep finding more and more, or wild plants. And even in fungi and bacteria, the OXO gene is very common. And it's a typical plant defense mechanism to fungal attack. So the way the wheat gene has been inserted is to piggyback on a naturally occurring bacteria, Agrobacterium tumefaciens. So it is, you probably know, has the ability to invade the genome of other species, happens quite commonly in nature. And so the wheat gene then piggybacks on that. So that creates then what we call the transgenic chestnut, which is 99.99+% American. It retains all of the American genes, more than 35,000 American chestnut genes, +1. The blight tolerance from lab tests is that it's similar to the blight tolerance of the chestnut, even though, as I mentioned, it's a different approach. But the process begins as a clone. So much of the work and the work that we do here at UNE has been to move through generations of breeding to create genetic and geographical diversification. What's here on the right side of the screen is some of the lab experiments with the transgenic wheat gene chestnut in the center, subjected purposefully to the inserted fungal blight, see what happens. Here's the pure American without the gene, which dies within a few weeks. And the Chinese, when subjected to the blight, also survives. And so that's the kind of lab results that indicate that inserting the OXO gene works. And now what we're spending a lot of time on is doing the field version of these same kinds of experiments. So in cartoon form, here's what happens. So the fungal blight uses oxalic acid to attack its host. And normally that would kill the American chestnut tree here, which has no natural defense. Now with the extra enzyme of oxalate oxidase, it has a defense. And then the chemical off products are carbon dioxide and hydrogen peroxide in small amounts. So it's 35,000 plus genes that are retained plus one extra gene. And all of this then means it's genetic engineering, of course, and that means that all of our work is subject to regulatory control and oversight. So everything we do here at UNE is under permit. And in particular, our relationship is mainly with the USDA, who we request permits and get permits from, but also overseeing our work is EPA and FDA, who will also need to judge whether there should be deregulation. We're seeking deregulation right now. But in the meantime, under these years of permit, we've got quite a bit of work to do. And in particular, in the speed breeding environment where Virginia has been so crucial to our advances. So I'm going to turn over the story to Virginia right now and tell you guys about our speed breeding project.
Thank you so much, Tom. That was a great introduction. So, why are we doing speed breeding? It's mainly because that in the wild and orchard, chestnuts don't mature until 4+ years to produce pollen. So we are cutting this time immensely. And by doing this, we expedite this process by seedlings to produce indoors year-round. So you can see some of our success that we've used two methods, which is on the left, which is going to be the transgenic pollen in the growth chamber versus what we've grown in the greenhouse. So we have two different methods, but because we have achieved such success with speed breeding, we have become the American Chestnut Project's leading producer for the past 2 years of transgenic pollen. And so you can see in the back, that's Tom with one of our seedlings right in front of the growth chamber. And just as an example to show how small the pollen grains actually are, it's only 15 to 20 micrometers, which can produce these 100+ foot tall trees, which is amazing. And so our methods that we use are extended light exposure. We use even temperatures that keep going throughout the day and night. We keep a relative humidity in both the greenhouse and the growth chamber. And we use a more acidic water. Oh, that's the data. That's our ongoing data that we keep monitoring in our greenhouse right now. So we have access to it. And so we can constantly monitor it to make sure that our plants are in peak condition. We also we work with the plants, and so we work with what the trees like best. And so we actually reduce the pH of the water because they like a more acidic soil. We also use different kinds of fertilizers to keep up with the growth process. And we're still experimenting to see which pots are best for speed breeding, especially 1 gallon versus 2 gallon, which holds the most water and moisture for the plants versus what makes the roots grow more or less. So we're constantly experimenting with how we can make our methods better. And of course, our most important thing is to keep the pests and pathogens at bay, because that's what really affects the growing process. So in this is a kind of temporal pattern that we have of our pollen production. On the x-axis, it's days since the nut has been like sown or planted. And then the y-axis is demonstrating how many slides of pollen have been produced. Slides of pollen we're going to talk about later and what that really means. And so you can see the slow maturation on the x-axis until about 277 days after sowing, it produces the first pollen. And then you see the exponential pollen increase. And then the roller coaster pattern comes from the decreases and then later increases. And these are caused by pests and pathogens. And the red arrows indicate like how we can make improvements in pest mitigation. And so our decrease in pollen on the last slide, it was caused by these pests and pathogens, which produce a unique year-round problem. So we have to deal with aphids, spider mites, mealybugs, powdery mildew fungus, and fungal gnats. So we use many different methods of pest mitigation, and we constantly have to monitor our plants like continuously, almost every day, for these to make sure there's no infestations. And as you can see, even though how tiny these pests are, they can make a very large impact. So why is it so important to mitigate pests? Well, we need to keep our producing high yields of pollen for each seedling. So you can see in this one, the pollen is hand collected on microscope slides. Like in our graph earlier, it was how many slides of pollen are produced. We use these glass microscope slides to collect and store pollen. And so after the pollen is collected, 4 microscope slides can be put into a labeled plastic vial, desiccated to remove moisture from the specimen, and then deep freeze at a negative 80 degrees Celsius in our special freezers. And so this method our team has produced has made enough pollen so we can be the main supplier of the chestnut project. And our research has produced over 850 vials, which allows this project to move forward exponentially quicker than if we had to rely on regular wild sourced pollen or just regular pollen grown in a lab, not sped bred. So this is a total of all our pollen slides produced at UNE. So each source is labeled on the x-axis on the bottom, and then its slides that it's been able to produce of pollen is measured on the y-axis. And so you can see that there's a high variability in the amount of slides that's produced per source. So some of them, like MA317 and WR30, are able to produce like over 50% of all of the pollen, and while others have not produced any. So this is a high variability that we have, and that's because of the individual plant genetics. Okay, and this is another photo of our process of the desiccation and freezing process of our pollen. And so our deep freeze allows us to store viable pollen and ship it across the eastern US to other branches of the project. And so this is actually a microscopic photo of our pollen grains that we were testing for their viability. So you see the pollen grains and that we're able to fertilize wild flowers. That means they're still viable. And when they're put in this agar solution that mimics wild flowers, they produce pollen tubes, which show their viability. And so through our research, we have been able to show that frozen pollen is still viable up to a year, for at least a year. And so this is another more microscopic photos of pollen grains in agar solution. And the photo has been edited to show the grains that are colored green produce pollen tubes and are viable, while the red ones did not produce pollen tubes, and so they would not be viable and not fertile. Okay, so this is a very heavy in-depth diagram that shows what we call the chestnut year. So from pollination of wild trees with our lab-grown pollen, to the sowing process, to the inevitable generation of the next new generation of pollen. So this is a sped up natural process of the chestnut maturation. We use speed breeding, which you can see is number 6 and 7, which is to produce pollen in record time, advancing our generation of offspring. So this is adding a crucial layer of genetic diversification, like Tom was talking about, because it's from a clone. And so we need more generations like out from the clone to diversify the genetics. And so this is our proof of methods and how successful we are, because this is a pollen source, which is produced pollen 6 months from sowing, like the actual planting in the ground. Well, something like this in the wild would take like up to normally 6 years. So where are we now? So we're at step 5 back in the chestnut year, which is the planting and sowing process. So in our greenhouse, we have about 60 diverse transgenic chestnuts that we're planting. And so these will be carefully monitored and grown in an attempt to produce the 4th and 5th generations of crosses that we can use this summer out in the wild to produce more pollen. So this is what I'm going to be doing for the next couple months. I'm very excited to be working in the greenhouse with them. So thanks so much. Back to Tom.
Great job, Virginia. Really nice overview of the speed breeding process. So now we'll turn briefly to talk about, well, what do we do with this lab-grown pollen on microscope slides that Virginia described? So we take it out to the field, and in the middle of July, when the female flowers on the chestnut trees are ready for fertilization, we apply it. And that's what's going on in this picture. And we've been doing this with 4 wild trees in a certain permitted site in Maine over the last couple of years to produce our crosses. So one of the questions we have, and certainly a really important issue for USDA, our regulator, is when we're bringing this pollen to the field that has this extra gene, so it's genetically engineered, does it drift? Pollen naturally flies through the air and can go 1 kilometer in distance from one tree to another. It's worth noting that chestnuts are bisexual, but they don't pollinate themselves. So this pollen will not pollinate this particular flower, even though it's on the same tree. So you need 2 to tango. So what we've done then is tested whether this pollen drifts like it does in nature. And what we found is happily 0 pollen drift off of our slides to flowers that are not our target, that are not inside of our controlled pollination bags. So that provides confidence that our method is very precise and confined. So this is what it looks like. The fun part, well, all this stuff is fun, but it's really fun to go up in the lift and do our pollination. There you see the pollination bags, crossing our pollen with the wild flowers on these trees. And again, the genomics are very interesting. We're learning things regularly from genomic analysis that we didn't know. And what it has recently revealed is that these wild trees that we pollinate with our lab-grown pollen actually have a significant amount of European species genes, anywhere, depending on which tree we're talking about, somewhere between 20 and 50% of their genome comes over from the European species through the importation that happened many decades ago. And the landowners we work with have no idea how that happened, but that's typical kind of hybridization that you find once you can look at it very carefully genomically. So the other thing we're doing in the field is we have these orchards all over Maine, and in fact, throughout the native range that other people are responsible for, but the ones we're responsible for in Maine are typically 100 sapling orchards that have tremendous genetic diversity. So they're representing the wild trees throughout New England, as well as other parts of the native range. And so we're doing this sort of genetic and geographical diversification. And so we're beginning to see some breakthrough there as these saplings, who in this picture are just 4 years old or 5 years old, are beginning to flower, and we can cross them again to create offspring that are blight tolerant, but also represent the native range. So back to this cartoon, and the crucial part of this picture here is the hydrogen peroxide that is created when the fungal blight's oxalic acid meets the oxo gene that's been inserted in our transgenic chestnut here on the right. This is crucial for our lab analysis of whether the gene has been inherited. So we do cotyledon sample testing in our lab to determine whether a particular chestnut has the gene. It's only on one half of the chromosome. And so half of the pollen grains inherit the gene, half of them do not. And so we can do a test then by taking a sample of the cotyledon, which is the meaty bottom part of the nut, and put it in a reagent that contains the oxalic acid, which is the killer that's used by the fungal blight. And we can test whether it has the gene. If it has the gene, it turns the reagent solution black from the hydrogen peroxide. So this sample here on the left has the gene, and the one on the right did not inherit the gene. This is still useful, the one on the right, because we do some full sibling analysis in the field. We want to see how the two perform relative to each other. But that's a really clean way, very precise way to find out whether the gene is inherited. And it's very precise in, with all the work that you do at Cornell with breeding. This is a really cool thing because we know exactly who the dad is. Because if it has the gene, we know through our pollination program and our controlled pollination bags, we know who the dad is. And we know who the dad is on this side as well, because again, because of the restricted, the inability of wild pollen to reach inside the bag. So you might wonder, well, can we chop up the nut in this way and take a piece out of the bottom of the nut, the cotyledon tissue, and still have a viable nut? So we've tested that over time, and we find that this method of slicing the nut very carefully, just a minimal piece, does not affect the germination. So we get germination rates in our greenhouse that are very similar to what you'd have a chestnut that was not sampled in this method. So here's what our chestnuts that have the extra gene look like in our greenhouse. This is what we do between February and May. We grow them indoors, and we try to get them to be basically 1 year size, the equivalent of what they would be like in the field after 1 year. But by May, we can have them that size thanks to the greenhouse method. So you can see they're quite happy. Here's about 400 chestnut seedlings that have inherited the trans gene here in one of our greenhouses. And then we've been out planting them. So we have that site you saw in the video in Cape Elizabeth, where we have about 500 seedlings planted that include the ones that have inherited the gene, but also their full sibling counterparts. Also, as well in this field comparison, we have 100% Chinese seedlings. We have the F1s, which are the 50-50 hybrids, and we have some that are advanced hybrids, like I showed you in that poster, that have 90 more ballpark, 93% American, 7% Chinese. And we're doing a controlled analysis of performance and growth in the field over time to see how indeed the transgenic approach compares to the other possibilities. So over time then, we've been monitoring them in the field, removing the protective tubes that allowed us to have a kind of a hardening process in the field, and everything was going hunky-dory in the field. But then, and there are always setbacks. We met, Virginia talked about the setbacks we have in long-term breeding indoors with the pests and pathogens. Well, we got pests in the field as well. So suddenly, out of nowhere, seemingly in August of this year, we had a big infestation of this. What's causing it? Look at the way that the seedling stems have been girdled, and it turned out to be mice that have invaded the orchard. So we had to deal with that. So there are various setbacks, and that's of course common in science: 2 steps forward, 1 step back. So that's what's happening in the field. It's again governed by permit. We're seeking deregulation, which might happen within 2 years, and that's going to be a very different part of the project when we're allowed to share our blight tolerant seedlings with others throughout the native range. But in the meantime, we've got plenty of work to do under permit. And so here's what our orchard looks like in kind of the most recent stage of growth. You can see some of our seedlings are tall. Some of them reach 4 feet tall in the first year, which would be more like a 2-year-old chestnut growing in the wild. in full sunlight. Another interesting experiment, I think, is that one of our seedlings that was a big pollen producer in the lab, we've been able to move out to the field and plant and keep happy as a a seasonal kind of seedling. And so we're hoping that this particular seedling, which has already had the genes turned on for pollen production inside, will do the same in the field in short order in order to provide another source of pollen that would be much easier for our project to be able to harvest in field setting. So the goal of then the field orchard over the next 5 years is to compare the relative performance of the transgenic seedlings that have the extra gene. How do they hold up? How do they grow? What's the cost of having resources devoted to producing oxalate oxidase compared to ones that don't? How do they withstand the fungal blight, which is ubiquitous, and eventually we will subject the seedlings purposefully to the fungal blight to see how they do. We're also interested in this orchard becoming a self-reproducing system of pollen and female flowers within the transgenic community, and that will eventually then produce some homozygous transgenics that have the gene on both sides of the chromosome. And so that will be a great breakthrough when we get to that stage. And then also just, again, waiting and doing what we can in the permit process to await deregulation. So eventually we're going to have saplings that are substantial at the rate they're growing. And these will be then some of the first saplings that have the gene, that have blight tolerance, that we can distribute by digging them up and moving them to publicly available spots, places where the public can see what we're up to, what they look like, and we begin to the very long-term, multi-decades process of repopulating the eastern forest with a tree that won't die of the fungal blight. We thank you for your attention, and we're interested in the subsequent discussion and questions. So thank you so much. All yours, Moira.
Thank you. Thank you so much. That was a very, I loved all the pictures. I loved the flyover of your orchard nursery, and you've done some beautiful work. It's very exciting to see. We're getting some questions in the chat. While people are formulating them, I have a couple of questions. I wanted to ask if you had to do anything to get the pollen to stick to the slides. So do you have to put any kind of like adhesive or treat the slides? This pollen is just sticky enough to adhere to the slide?
It is sticky, right? It is
Okay.
sticky, so it adheres quite easily. Yeah, there's a certain method of sort of tapping the catkin, which is where the pollen resides. But yeah, it sticks really nicely to the slide.
Okay. My second question is, what promoter was used for the transgene? So, you know, promoters can be very tissue specific, but you're seeing expression even in the nut. So it's probably either under some over-regulation or ubiquitous promoter.
Yeah, it's a good question. And there's so many different aspects of this project we didn't have a chance to touch base on. But the current promoter leaves the oxalate oxidase on all the time. So it's like a fire hydrant approach. And so that's one of our questions of how much that matters, because even if the plant is not being attacked by oxalic acid, it's producing and using resources to produce oxalate oxidase to protect itself. So the newer version of the promoter is what's called a wound-induced promoter. Mm-hmm. And that then is turned on only when it finds the oxalic acid coming its way. So that's a more efficient method. That's a newer method. We don't have pollen from that yet. We hope to have that pollen this summer. And then that'll be also an additional important comparison. Does it matter? It may not matter. It seems like our on all the time promoter still grows trees very vigorously, not only in our early experiment, but the other work that's been done at ESF. The plants seem to grow quite quite tall and vigorously under those conditions, but that's certainly something we're going to look at into the future. That's the same, since it's the same transgenic insertion, it's governed by the same permits. So the promoter itself is not something that USDA has much concerned about. It's the genetic engineering that really.
Okay. Excellent. All right, so we have a couple of questions in the chat. One is from Bruce Reich. What are the prospects for oxo resistance being long-lasting, durable resistance? Are there strains of blight that may overcome the resistance?
Want to make any, Virginia, you can jump in at any point if
Yeah.
you'd like, but let me know if you want me to handle them. What do you think?
I think you can handle this one, like the long lasting,
Yep.
but from what we've seen is the fact that it always is producing this, and I haven't seen the resistance. It acts similarly, like we said, to the resistance that the Chinese chestnut has, which is it co-evolved with it, so it's able to kind of balance it out. But Tom can probably talk on the long-lasting side.
Yeah, that's really important. And the other thing about it's a great question is that our method does not kill the fungal blight. Okay, so it's not encouraged to mutate in order to get around. It's not a pesticide. It's only keeping the chestnut tree itself from being killed. But what we know from the Asian context, and even in the American context, because the Cryphonectria parasitica reproduces on oaks, it doesn't kill oaks, but it reproduces on them. So it, again, mentioned before, it's happy to function as a saprotroph, which basically means it's consuming dead material from other sources. So it need not act as a necrotroph to kill the American chestnut. So we feel that that provides a long-term solution because of the discouragement of evolving, mutating around the defense.
Okay. There's another question from Bruce. Are there any prospects for nut production in the greenhouse? It is an impressive gain to obtain pollen in 6 to 12 months, but can these trees also set fruit?
Yeah, I can also talk on this because I was highly involved in the pollen production, especially last year. So what we're trying to do is we almost treat them like tropical plants. So we leave the larger wild trees to produce like the female flowers that we use for pollination and fruit bearing. That's not something that we're able to achieve in the lab because you need such like a massive tree because it takes longer to produce the fruit and the flowers that come with it. So what we've been focusing on is just the pollen source, which is already hard enough to do in the lab, and we're going to let the natural process in the wild take that side of the coin. So we're just here to produce the pollen, and it would be incredible to make, you know, nuts in the lab, but we want to use the natural and wild genetics to even more diversification in the future.
Yeah, and those rare occasions when we do have female flowers on our seedlings, we get all excited about it. They inevitably cannot reach maturation because there just isn't enough resources. It's a completely different ball game on the female side. So that's where we rely on either the mature trees or the saplings in the field for the female side of the equation.
Great. Okay, another question from Jeff Doyle. You're propagating chestnuts that are F1 or later generations, hybrids between European C. sativa and North American C. dentata, and the plan is to repopulate the eastern U.S. with these trees rather than with genetically authentic C. dentata. Does that bother you? And what are the ecological or even bioethical implications? And are there no pure C. dentata that could be used?
Yeah, there are, and that's where that one slide of the germplasm conservation orchard, where we're beginning to pollinate those saplings with our transgenic pollen. Those are 100% pure American. So there's not just one outcome. There's lots of different types of crosses. In fact, we produced 39 unique crosses this summer between various trees and various the variety of different pollens. So we're not relying by any means strictly on the ones that have some sativa genes. The thing about the sativa, which is the European species, is that it's the most similar to the American. It also grows as a tall canopy tree. So phenotypically, it's very hard to tell the difference between the two. And that's why we were a little bit fooled until we had the genomics. But the main tree that we're using in the field, in fact, has about 20% sativa genes. So immediately then when you do a cross, you're down to 10%. And then when we're crossing again, as we were doing, you're down to 5% European. So it's a pretty small percentage. And remember, the other approach, the breeding approach with the Chinese, is you're needing probably at least 10% Chinese in order to have, and likely more than that, more like probably 20% Chinese in order to have enough of the genes transferred over to provide blight tolerance through the traditional breeding method. These are good questions.
Yeah, they're great questions. I can kind of also touch on the bioethical implications, which is, what was it? ESF did a huge amount of research with how our transgenic chestnuts function in wild environments. And so it actually showed that they behave the same way that the non-transgenic pure American chestnuts would. So it still had the same effects. It didn't affect pollinators or other organisms that fed on the leaf litter or anything. Actually, tadpoles grew faster while eating the transgenic leaf litter, or just, in general, opposed to like if they ate oak or maple, which are the replacement trees that came in after the chestnuts were wiped out.
Let me add one other thing, because it's a really great question from Jeff. Is that, and this kind of gets back to why we have this problem in the first place with these chestnut species being brought into the United States from other parts of the world. The American chestnut is the smallest size nut of all the different options. And so back, more than 100 years ago, people were bringing these other chestnuts in because they produce larger chestnuts for domestic food consumption. So actually, one of the advantages of having some sativa genes is that it produces a larger nut. So the goal of the restoration certainly is repopulating the forest, but also is to provide human food. And so a chestnut that has some European genes actually produces a larger nut, which is more valuable in terms of the human consumption side of the demand. So there's going to be a lot of different demands. And certainly if we're interested in repopulating the forest for wild animal production, we'd want a pure American because of smaller nuts. If you want it for food production for humans, you'd want a slightly larger nut.
Jeff, did you have another question? I see that you came on your camera, so I didn't know if you had an additional question you wanted to ask before we go on to Tim's question.
Just wanted people to know I was actually here.
Good to see you.
Good question. Yeah, thanks.
Thank you very much.
Okay, we have a question from Tim Setter to Virginia. Your method of growing plants in the growth chamber is intriguing. Please elaborate on what you mean by saying you use blue light LEDs, for leaves and red light for flowers.
Okay, so Tom has also been doing a large amount of research on this, and we've had a lot of help with indoor plant growing and how different kinds of like wavelengths of light can help them grow better. So it's like, as you saw some in our greenhouse photo, when there was about 400 of them there, some of their leaves still had the red spots on them. So that means that they were still maturing and growing. So they could, the blue light is better for the leaves, for them to absorb it. Correct me if I'm wrong, Tom. So you can also talk on this. And so the red light for flowers, you said?
Induction.
Yes. So that is for when we were growing them in the later stages of, we use like kind of, it almost looks like a pink light. So you go in and everything's lit up and it's pink. And it helps promote the catkin growth, I'm pretty sure. So, which is really cool to see, which is produces the pollen. So I'm not exactly sure entirely how it does it, but we've learned a lot from other people that have been doing a lot of like indoor growing and like
especially the marijuana growers.
Yeah.
So they are the real experts on all of this. So we just bought some lights that they say are the absolute best ones. Super expensive, more because the price of all this has gone down dramatically. But we would just put one in our greenhouse that they say is the Cadillac, as we used to say, of LED lights that has just the right spectrum to grow your plants most efficiently. So we'll
They've
see how
been
that turns out.
incredible. And we're using, in one of the last pictures of our greenhouse now, we have the heating pads almost to keep the chestnuts warm, so their soil and everything doesn't get too cool and they can still maintain the right humidity and temperature, which was another addition from the marijuana growers. They're awesome.
I have another question. Given the importance of American chestnut to indigenous people, are you doing any kind of outreach or DEI work with any indigenous tribes in the area, or do we know of anybody who is doing that in the American chestnut community?
Yeah, great question. Really important question, because this tree was important for literally thousands of years to Native American people. So yes, there's a lot of conversations going on of that sort throughout the Native range with Native American people as to what their interests are, their desires are. The certain goal from our side would be to provide chestnuts to any Native American people that would be interested. The big breakthrough on that, it was a memo of understanding that was signed about a year ago between the American Chestnut Foundation, which we're part of, and the Eastern Band of the Cherokee Indians in the sort of the heart of the range. And so that was the result of a long-term conversation, which of course is the appropriate way to go, and talking about the different interests and questions that both sides would have. So that was the big breakthrough, very exciting, but it's certainly really a priority that any Native American people that would be interested would have access to trees that can survive the fungal blight.
Especially in Maine, we're watching, actually, we were doing genetic testing and we've been watching and following the news on different land back movements, especially in Maine, and the different legal proceedings that are happening right now.
Excellent. Any other questions? Anybody who's on the call is welcome to unmute themselves or raise their hand. I see Lee has a comment. Charlie Burnham would be proud of you all. Thank you for the seminar. Anybody else have any additional questions for Tom and Virginia?
And just so everybody knows, when you hear the word Burnham, that whole plan of breeding the chestnuts that we laid out briefly there, that was the Burnham plan. So tons and tons of decades-long arduous work are behind that, and really catapulted us to the point where we are now. So yes, thank you, Charlie Burnham.
Excellent. Okay. Well, I want
to speak up themselves. Feel free to just chime in verbally. Go ahead, Moira. I didn't mean to interrupt.
Oh no, I don't see anybody else with their hand up. I don't see any new chats coming through. So
I would just be interested in try to throw a question back your way, Moira, or to anyone else. Give us a feel. We're out here in the northeastern tip of the native range of chestnuts, doing our thing. What did you hear today that connects in any way, shape, or form with the kind of work that's going on in Cornell? And we regret, obviously, with the COVID restrictions, we're not right there rubbing shoulders with you and finding out about your work. But I'd love any comment from anyone in the audience about the kind of work you're doing that has some connection to what we talked about today.
I could certainly go. I'm as Breeding Insights working with several tree species now. The promise of genomic selection and speed breeding really opens up a huge avenue for improving citrus for HLB tolerance and maintaining the American and Florida and California citrus groves in perpetuity, or at least being able to adapt more quickly to new invasive species. And certainly pecan would be another one that we would benefit. But I also see applications that could possibly be used in other slow growing perennials. So something more in the shrub and vine area too, that we may be able to do some speed breeding for too. And that would really allow us to change how quickly we can make genetic improvements in those species, rather than, waiting 5 years for citrus to flower, making one cross, waiting 5 more years for the next one to flower. We could really get more more cycles in much quicker. And so the potential is really big for me to take this technology and try to make it work for other species. Anybody else want to comment on the impact that, anything that touched them about this? Marvin?
Yeah, so I have a graduate student, Samantha Bosco, and she's developing a model for chestnut growth, looking at biomass accumulation, carbon accumulation over time, and also looking at tree establishment. And she's also done a lot of work with some indigenous communities and some survey work here in New York with the Seneca Nation.
Awesome.
Yeah, we'd be very interested in that. We should, if you maybe can put us in touch with Samantha, that would be fabulous.
Yep, will do.
Yeah, good stuff. Yeah, really important. Yeah, it's a fast-growing tree. It's a carbon sequestrating tree. The chestnut is deep tap roots, so it survives the windstorms. We've had white pine trees fall on our big chestnut trees and break off parts of them, but the chestnut tree remains standing, just kind of showing the contrast in the structure of the tree. It's pretty phenomenal. Yeah, so good soil enhancing tree, symbiotic mycorrhizal tree.
I think there's one more question in the
There
chat
is.
that was could hydrogen peroxide be a problem for the transgenic plants?
Okay, so that's when we, no, I mean, hydrogen peroxide is very common in nature. It's all over. And it's this is produced in very tiny amounts. So it doesn't really pose any negative impact. We don't see any problem whatsoever with the production of that as the oxalic acid is broken down when the tree is under attack.
So
anybody else have any questions or comments they'd like to, unmute themselves or turn on their video? Okay. Well, then we'll let you, Tom and Virginia, go and have a great holiday season. And thank you so much for joining us and for being able to do this via Zoom. As we mentioned earlier, that we went into red status, so all in-person things were canceled. So I'm so glad that this didn't happen with you on campus, and we still got to have a nice interaction with you today. So thank you so much.
Best. Yeah, thank you so much. I know you're busy with finals and everything at the end of the semester. Thanks for taking the time out to hear what we're up to over here, and wish you all the best with the COVID outbreak. So yeah, great. Thank you. It's been a pleasure.
Thank you so much.
Thank you so much.
All right. Bye-bye. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.